Welcome everyone. Greetings from the Australian National University in Canberra. My name is Jay and I look after international relations and partnerships here at the ANU for the disciplines of sciences, health and medicine. I'm joined today by my colleague Arthur Xu and of course our speaker for today, Dr. Anna Herring. I'd like to begin by acknowledging the Ngunnawal and Ngambri people who are the traditional owners of the land where ANU is located and where we are broadcasting to you today from. And I pay my respects to their elders past and present. Let me now briefly introduce our speaker for today, Dr. Anna Herring. Anna is currently a research fellow at the Department of Applied Mathematics in the ANU Research School of Physics. She is also a chief investigator for the Australian Research Council Training Centre for Multi-Scale 3D Imaging, Modelling and Manufacturing here at the ANU. Anna's research is largely experimental and is based on highly resolved visualization of fluid, gas and liquid flows within permeable media. Her research interests focus on microscale physics and fluid porous media systems with applications in carbon dioxide storage and carbon dioxide utilization. She is known for her novel characterization approach to utilizing topological analysis of 3D image data, and her research enables efficient design and optimization of technologies used to fight climate change, such as storing carbon dioxide in permeable underground geological formations and in cements used in construction. As a testament to Anna's research, in 2018, she was awarded the Australian Research Council Discovery Early Career Research Award. So without further ado, I'll now hand over to Dr. Anna Herring to tell us more about safe and efficient carbon dioxide injection and storage strategies. Over to you, Anna.
Great, thank you so much, Jay, for the introduction. Very nice. I'll go ahead and share my screen. All right, so yeah, my name is Anna and I'm going to talk to you today about storing carbon dioxide in rocks. Okay, so I'm going to start with the really depressing stuff first. It's become increasingly obvious in the past couple of years that we're not currently doing nearly enough to limit the worst impacts of climate change. There are lots of good scientific reports out there on why it's crucially important to limit warming. In particular, I'm going to point to this IPCC report from 2018, as it goes over the really wide range of really severe impacts that we can expect to see even at 1.5 degrees of global warming. But the main message here is that it's not looking too good that we'll be able to achieve the goal of limiting warming to 1.5 degrees Celsius. And if we do indeed want to avoid the worst impacts of climate change, it's going to require large-scale decarbonization on a massive scale worldwide. Okay, we know that climate change is overwhelmingly due to anthropogenic emissions of greenhouse gases. And so in that framework, there are two related but separate ways that we can think about addressing emissions. So I'm going to be talking about both of these throughout the first part of this talk, but I just want to make sure that the distinction between them is clear. So emissions mitigation means reducing our current emissions. Let me get rid of my laser pointer. So for example, we can think about increasing the efficiency of our electricity generation and transport systems, increasing fuel efficiency for vehicles, switching to low or zero emissions electricity generation or industrial product generation techniques. We can switch to low or zero emissions vehicles. We can do all sorts of things with our personal decisions that minimize our personal carbon footprints, so on and so forth. The idea here, though, is that mitigation is basically just reducing our current emissions. Negative emissions, on the other hand, can use some of the same technologies as mitigation technologies, but they're distinct because a negative emissions technology involves a process that consumes more carbon dioxide than it emits. And keeping in theme of the, disappointing or depressing aspect of this part of the talk, it's become obvious that we're going to need to start implementing negative emissions technology if we want to keep global warming below 2 degrees Celsius. Okay, so here I'm showing a figure. This is from the IPCC report that I pointed to earlier. And I really like this figure because it sort of shows the relationship between emissions mitigation and also negative emissions. So here what we have are 4 different scenarios that the IPCC has addressed in this report. The P1 scenario is a very optimistic scenario, and then it ranges upwards to P4, which is a scenario where we move much slower in confronting climate change. And basically, anything that happens above this 0 line is relating to emissions mitigation, and anything that happens below the 0 line is relating to negative emissions. So basically, this is telling us that if the shape of the emissions mitigation pathway looks like this, above the line, we're going to need to do this much negative emissions in order to accommodate the continued use or the continued CO2 emissions if we still want to be able to limit warming to 1.5 degrees Celsius. So in this really optimistic scenario, we see that if we can have emissions from fossil fuel and industry peaking very soon and then dropping very rapidly, we can get away with a pretty low level of negative emissions. And actually that could be accomplished entirely with land use changes. So in this figure, the brown color is indicating land use changes. On the other hand, if global emissions peak a bit later and then also don't drop as fast, we're going to need to implement much more significant levels of negative emissions technologies. And we can no longer rely just on land use change. We need to start deploying more advanced engineering-based technologies, and that's what's indicated in that yellow color. Okay, so the takeaway here is that if we want to limit warming to something like 1.5 or 2 degrees Celsius, it's not good enough to do better and reduce our emissions. It's not even good enough to just stop emitting entirely. We are actually going to need to start removing CO2 from the atmosphere via land use changes or some other technologies. And eventually we're going to need to do something with all of the carbon dioxide that we're removing. And so this paper that I had shown a bit earlier shows it actually provides 2 scenarios with some numbers. So it shows that in the best case scenario that was investigated in this paper, we're looking at on the order of 0.5 gigaton of carbon per year needs to be consumed by a negative emissions process. And when they forecast that over 100 years, that amounts to 50 gigatons of carbon total. In the worst case scenario that they looked at, that number goes up to 11 gigatons of carbon per year, and then 1,600 over the 100-year forecast. Okay, so how much carbon is that? I think that sometimes it can be really hard to understand what these numbers mean. Like what is a gigaton of carbon actually? And so as a comparison, here I'm showing a figure of terrestrial just above ground biomass in the Amazon rainforest. So this is all of the trees and shrubs and all the flora that's above ground in the Amazon rainforest, and that comprises about 76 gigatons of carbon. So when we compare that to these best and worst case scenarios, we can see that if we're on the best case scenario, maybe over the course of 100 years, we could build approximately 2/3 of an Amazon rainforest, and that would provide enough negative emissions to accommodate that best case scenario. But in the worst case scenario, we're going to need something like 21 times the amount of carbon that's stored in the Amazon rainforest. Okay, so what else can we do with carbon dioxide? So I've already sort of just mentioned land use changes. We can also try to utilize carbon dioxide. So I sort of think of this as reusing or recycling the carbon dioxide. So for example, potentially we could make carbon neutral liquid fuel from it, or we could put it into useful products like concrete or plastics that are then consumer products that people actually want to use and want to pay for and buy, and then use to make useful things. The other option that we have is to come up with some way to safely dispose of the CO2, which is the storage option. And so our main option here is geologic sequestration. And I have these asterisks here beside all of the utilization and storage options because I really want to emphasize that I'm not proposing that we do these things in place of continuing to do research and development and deployment of renewable technologies. We actually have to do this in addition to all of these other things that we need to do to increase efficiency and find better sources of energy. So this is something that we need to do in combination with other research into finding better ways to produce energy and make products. Okay, so geologic sequestration. The idea is that we are going to capture CO2 as a gas and pressurize it until it's roughly the density of a liquid, pump it deep underground. We're targeting formations that are at least 800 meters deep, but often more than 1 kilometer deep. And what we would really like to do is find formations that have layers like this. So often sedimentary formations have layers of really high permeability, high porosity layers bounded by layers of low permeability, low porosity. And the idea is that we're going to inject the CO2 into the high permeability, high porosity layers because there's lots of storage capacity in those layers, but then the CO2 is going to be bounded by these impermeable layers. Coming back to this idea of emissions mitigation versus negative emissions. So CO2 sequestration can be an emissions mitigation technique when it's used to reduce or remove carbon emissions from, for example, fossil fuel production, electricity production, or from cement production plants or other industrial processes that are emitting CO2. It could also be thought of as a negative emissions technique by using the same technology for the geologic sequestration part, but when we capture the CO2 directly from the atmosphere. So for example, via direct air capture or via something like bioenergy, carbon capture and storage. And so I'll just briefly explain what that is. Bioenergy, carbon capture and storage is this idea that maybe we'll plant large plantations of trees, and then we can harvest the trees, combust them to get energy from the trees. But when we combust the trees, we capture the carbon and then again pump it underground. So the idea here is that we use the trees as our carbon capture technique, and then but then go through the same process to pump the CO2 underground. Okay, so as a case study, I wanted to introduce you all to the Sleipner project, which is in Norway. So they've been injecting CO2 into this formation since 1996. That's because Norway has had a carbon emissions tax since the early 90s. And they had an oil field out here. The oil that they produced had elevated levels of CO2, and they needed to remove some of the CO2 before they could sell the oil to consumers. So basically, they're pulling the oil out of this lower reservoir. They strip off the CO2, and then they re-inject the CO2 into this layer that's sitting above the oil reservoir. And so they've been sequestering almost a megaton of CO2 per year in this project, which is really cool. Here's just to put it in context. So of course, here's the world. Here's Norway. This box is roughly the same size as this box. And then it's this formation that's out here in the North Sea where they're actually doing both the oil production and the CO2 sequestration. And most of the pumping occurs in the southern part of that field. And since they've been doing this for almost 25 years, they have a lot of really good data on how the CO2 is moving underground once they pump it under. So these are from 3D seismic monitoring studies. So here's a Z dimension profile, and you can sort of see before the CO2 injection started, there's this layer here at the top. As the CO2 injection is progressing, we can see that the CO2 is building up underneath the layer, but not actually breaking through. And then when you look at that same region in a planar view, you can see that the CO2 plume is growing and spreading laterally, but again, not breaking through that upper layer. Okay, and so recently, the Norwegian government has decided that they want to expand this project even further. So instead of just injecting the CO2 that comes from the oil production facility, they also want to start sequestering carbon dioxide from other industrial sources. So this is a joint venture between the Norwegian government and industry partners. And in this case, they're going to be capturing CO2 emissions from a cement plant and also a waste to energy plant. And then they capture the CO2 at those point sources, put it on a boat, transport it to this distribution point, and then it gets piped offshore to the injection site. And they're actually building the infrastructure to handle 10 times more than the current cement and waste to energy partners will produce. So they have room to grow. And they're estimating that they'll be able to sequester 5 megatons of carbon dioxide per year. And this is just some other potential sources. As they are growing, they would potentially be able to start accepting carbon dioxide from more industry partners throughout Europe, which would be a really interesting business plan. Okay, so I want to just walk through the question though of how much CO2 is the Northern Lights project going to be storing relative to what we know is necessary? So I just want to walk through a quick example of calculation comparing that 5 megatons of CO2 per year to the values from this 2015 paper. All right, so the first thing that we need to do is convert megatons of carbon dioxide to megatons of carbon, because we know that those two are not directly equivalent. So first we have to do some basic stoichiometry to convert megatons of carbon dioxide to moles of carbon dioxide. Then we can say, all right, we know there's 1 mole of carbon in every mole of carbon dioxide. And then we can reconvert that back out to mass. And we find that the Northern Lights project is storing approximately 1.4 megatons of carbon per year. Then we can convert, of course, megatons to gigatons. So that's a factor of 1,000. And then we can finally compare to the numbers from that 2015 paper that I referenced. And we can see that for both cases, we're much lower than 1% of what's going to be needed to be sequestered. So I guess I just wanted to walk through this calculation to sort of give people a sense of the scale of the problem that we're running up against. So of course, the Northern Lights project is not the only carbon dioxide storage project that's operating worldwide. So there are more storage facilities, and there's even more in development. So this is a figure from the International Energy Agency. The blue bars are storage projects that are either operating or under construction, so they're soon to be in operation. The green bars are storage projects that are in various stages of development. And so it's possible that some of the projects in the green bars will never actually make it to the blue bars. And so what this is showing us is that we currently have the capacity to store 40 megatons of carbon dioxide per year on a worldwide basis over all of these projects. Okay, but again, we can compare this to what we know is needed, and we can still see that currently we're less than 1% of what we know is going to be needed by 2050. Okay, so this is an image showing possible storage sites and capacities worldwide. So I think this shows that there is potential here. We have the storage capacity that's needed to meet the demand for storage. But the problem is that we still need to actually develop these sites. So for example, the IEA says that we need to develop 30 to 60 storage sites every year until 2050. And that's only from a mitigation standpoint. So if we want to include additional storage to account for negative emissions, those numbers could double. They do point out that this is feasible. So you can compare to oil and gas development in the first decade of this century. But this is still going to be a heavy lift. So we need to get going with developing these sites. Okay. So now I think I've explained why I think this is such a crucial technology and something that's really important to study. I want to talk to you more about what actually happens during CO2 sequestration. So once the CO2 is injected underground, it's still buoyant relative to the resident formation water. So it's going to buoyantly migrate upwards. And it could eventually make it back to the surface unless it's securely trapped underground. So there are several trapping mechanisms that we need to study and understand. First one is structural trapping. So as I mentioned previously, we're trying to find layered systems where we can inject the CO2, and that ensures that there's an impermeable cap rock on top of the CO2 that helps to hold it just physically down. Another physical mechanism is capillary trapping, or it's sometimes called residual trapping. And this is basically as the CO2 plume is moving through porous rock structure, some little tiny bubbles of CO2 get snapped off and trapped between the grains of the rocks, and they're held there due to capillary forces or interfacial forces. So that's capillary trapping. Solubility trapping occurs when CO2 starts to dissolve into the groundwater that's present in the storage formation. So water that has CO2 dissolved in it is more dense than water that doesn't have CO2 dissolved in it. So as the CO2 starts to dissolve into that resident water, the water which now has CO2 is more dense and will start to mobilize downwards, and it makes these really quite beautiful fingering patterns as it's moving downwards, and then fresh brine comes back up from the bottom and replaces it. So this is the solubility trapping mechanism. And then mineral trapping occurs when carbon dioxide undergoes chemical reactions with minerals in the subsurface, and then when the carbon dioxide itself mineralizes and makes carbonate rocks underground. And of course, when we're trying to predict what happens to CO2 when it's pumped underground and to design these sequestration projects in a way that's safe, we need to be able to quantify how much CO2 can and will be trapped by each of these mechanisms. We need to know what timescales that will occur on, and then how secure is the CO2 when it's trapped in that form. So in general, when you go down this list this way, or from left to right on this other figure, the storage security increases, but also the time that's required for these mechanisms to start playing a significant role in trapping also increases. And so as I said before, we mostly address structural trapping by trying to find good geologic storage formations with good layering. But for these other trapping mechanisms, we actually have opportunity to optimize these trapping mechanisms by playing with different injection parameters. So in our lab, we mostly focus on these middle two. Today, I'm mostly going to talk to you about capillary trapping in more detail. Okay, so to talk about capillary trapping, it's necessary to understand capillary pressure. So I'm going to go through some basics here. So the capillary pressure of a single bubble is equal to the pressure inside the bubble minus the pressure outside the bubble. And capillary pressure is proportional to the interfacial tension between these two bubbles, and it's inversely proportional to the radius of the bubble. Okay, so here's where we have our poll question. So I want to just see if people understand this. So we have a blue bubble, which is quite large, and a red bubble, which is quite small. These are intended to be the same fluid. I've just color labeled them to separate them. And they are floating within a second different immiscible fluid. But we will allow for mass transfer between the blue bubble and the red bubble. So I want to ask you the question, if the system is allowed to equilibrate, if mass transfer is allowed to happen, what will happen to the bubbles? Right, so the poll question is up. You have three options. You can have the blue bubble will diffuse, the molecules in the blue bubble will diffuse towards the red bubble until they're both the same size. The blue bubble, the molecules in the blue bubble could diffuse towards the red bubble until the blue bubble completely disappears and the red bubble grows, or you could have the opposite, the molecules in the red bubble diffuse to the blue bubble, to the point where the red bubble disappears, but the blue bubble grows.
You want me to give it a couple of seconds more, Anna?
Do you have some answers already?
Yes, about 60% of the people have voted.
Okay, I think that's probably good. So maybe, yeah, maybe we can stop the poll. Yeah. Okay, so the most popular answer is the blue bubble. The molecules in the blue bubble will diffuse towards the red bubble until both are the same size. So if we go back to the capillary pressure hole, or the capillary pressure equation, which I just showed you, what we know is that the capillary pressure is inversely proportional to the bubble radius. So actually, in this scenario where we have two bubbles of the same fluid but different sizes, the red bubble is at a higher pressure and the blue bubble is at a lower pressure. So what would happen in this case, theoretically, assuming you could set up this experiment, is that the red bubble would diffuse completely into the blue bubble, the red bubble would disappear, and the blue bubble would grow. So almost 30% of people got the correct answer, so that's really good. But okay, so we can go on to the next bit. Okay, so we can make this slightly more complicated now. So now we have a bubble in the presence of a solid. So we can modify the Young-Laplace equation to include the interfacial interactions between the solid and the two fluids. And we do that by including the contact angle. So the contact angle basically measures the affinity of the solid surface for one fluid relative to the other fluid. So for example, if you have two capillary tubes with the same radius, but with different contact angles, the capillary tube with the smaller contact angle will pull up water to a higher height. And in the same way that capillary tubes pull up water, hydrophilic rocks will also pull up water. So in order to push a non-wetting fluid like CO2 into a rock, you have to apply a higher pressure. So the CO2 pressure needs to be higher relative to the water pressure to actually force CO2 into the pores of the rock. Okay, so that's sort of what I'm trying to show with this cartoon. So now we have a non-uniform geometry. So this starts to approximate a pore space pathway through a porous rock. So it's more complicated again, but we can still use the Young-Laplace equation as an approximation to determine the threshold pressure of the constrictions in the rock. So the pressure threshold at this constriction in this pathway is going to be roughly equivalent to 2 times the interfacial tension between the two fluids, times the contact angle of the rock, then divided by the radius of the constriction. And that's assuming a cylindrical pore throat. And the capillary pressure across this interface is going to be equal to the pressure difference between the two fluids. So the CO2 pressure minus the water pressure. And we can measure this difference as 2 times the interfacial tension between the two fluids multiplied by the mean curvature of that interface. So essentially, if the capillary pressure is sufficiently high, so if the CO2 pressure is much higher than the water pressure, then that can force this interface to become more tightly curved, and that will allow the bubble to push past the constriction. On the other hand, if the capillary pressure across this interface isn't large enough to exceed this threshold pressure, then the bubble is going to stay trapped below that constriction. So what I'm trying to say is that in order to estimate how much CO2 is going to be trapped by capillarity in a rock core like this, we actually need to be able to look at the microscopic pathways through the pore spaces in the rock and measure the sizes of these constrictions. So this is actually an X-ray microscope image, a 3D X-ray microscope image that we took in our at the ANU CT lab. And you have rock grains, they appear as sort of this gray color, and then you can see the pore space, which is this darker color, and then we can also see some clays present in the rock as well. Okay, so this is what's called a pore scale perspective. So we're looking at these processes on the scale of individual grains and pores within these rock cores. And of course, this is in contrast to what we really care about, which is a reservoir scale or basin scale process. So this is actually the real scale of an engineered injection site, and this can be, tens of kilometers by tens of kilometers wide. But of course, the issue here, and this is a really complicated and tricky problem, is that these very, very small features that dictate capillary trapping inside the rock pores have a really significant impact on how the CO2 transports and becomes distributed on this larger scale. So I'm showing here results of some modeling studies that showed, so they did these large scale models looking at how CO2 moves after 500 years after injection has stopped. And if you don't include this capillary trapping process, then basically the CO2 is injected at all these different wells, and it all just migrates to the top of the reservoir and pools up there. On the other hand, if you do incorporate capillary trapping into your model, we can see that the CO2 is much more broadly dispersed, and it doesn't ever form, a gas cap underneath the cap rock there. So it is really important that we understand what's happening on these small scales and then try to incorporate it into our larger scale models. So of course, the way we do that at the ANU CT lab is with X-ray computed microtomography. So this is direct 3D imaging of internal structure using X-rays. I think people are probably familiar with radiographs. This is just a projection of X-rays through a sample. Tomography is similar, except for we're doing it in 3 dimensions. So we're taking projections all the way around the sample. And then from this, we can reconstruct a 3D model of X-ray attenuation within the sample, and you can start to look at slices through the sample. Here's just a schematic of how the experiments are set up. Basically, we have a sample on a sample stage. We rotate that sample while also translating it vertically. And as we're doing that, we acquire thousands of radiographs on a helical acquisition trajectory. And this allows us to get scans of quite tall samples and get good measurements of X-ray attenuation throughout the entirety of the sample. And then because we have these X-ray attenuation measurements from all angles, we can reconstruct the 3D distribution of X-ray attenuation at every point inside the sample. And then from that, we can determine the material distribution at every point inside the sample. Okay, so here's some videos from my colleague at the ANU CT lab, Andrew Kingston. This first video is showing the acquisition pattern of a Slater bug. So in Australia, they call it a Slater bug. I would have called it a roly-poly, but it's these little bugs that curl up into a ball. So they've scanned one of those. So this is just what it looks like as it's rotating inside the machine. This is a video which is trying to show you how the reconstruction works. So basically, if you start with just one angle, there's not very much information. And as you add in information from all the way around the sample, you can see that the image becomes fully reconstructed, in 3D. Oops. And then, so once we've gone through this process, we now basically have a 3D digital model of our sample. And so this is just a 3D visualization of that same Slater bug that I just showed you previously. Okay, so we use the same technology to look at CO2 and brine flows inside our rock samples. And to do that, we have designed, built, and implemented a high pressure flow cell. So in this case, let me grab my laser pointer again. So here's the X-ray source. The sample sits on the stage here, and here's the detector. And all of those imaging components are contained within inside a lead cabinet, so that protects all of us experimentalists from the X-ray radiation. And then outside we have pumps which maintain pore and confining pressure, in which we use to inject fluids into the rock. And everything is maintained at reservoir relevant pressure and temperature. Okay? And that becomes important because when we're injecting CO2 underground, pressure increases as you go underground due to the hydrostatic pressure gradient. And so when we do these operations, we're targeting depths such that the CO2 is forced into a really dense fluid phase, because in that way we can basically store much more CO2 than we could if the CO2 was in a gas phase. So we're targeting these quite deep layers so that we can get the CO2 into a dense phase. But when we get that deep into the subsurface, there's also a geothermal gradient, and so the temperature goes up correspondingly. So what happens is that when the CO2 gets injected, it is injected basically as a supercritical fluid. And so this means it's not a liquid or a gas, but it's sort of has characteristics of both. So it's quite dense and it flows like a liquid, but it's still compressible. And the other fluid properties also change pretty dramatically with pressure or temperature. So things like viscosity and interfacial tension also change if you would change pressure or temperature even slightly. So this means that for our experimental setup, everything needs to have the same pressure and temperature to keep everything, to keep that supercritical CO2 in the same phase and the same state. So this is a video I took just the other day of our flow cell. This is our fluid mixer. So here we're mixing CO2 and brine so that the two phases are equilibrated with each other. So this was at 1350 psi and 50 degrees Celsius. So here we have water that has potassium iodide in it. So that's called brine. So it's yellow because of the potassium iodide. And we have supercritical CO2 that's on top, just sitting on top of the brine, but mixing together. Okay. Yeah, so just to make that point again, everything in the system is temperature and pressure controlled to keep those conditions consistent. Okay, so I'll just quickly go through the experimental process. This is a 2D slice from a 3-dimensional data set. So we're looking at the internal cross-section of a rock. The gray values in this image represent X-ray attenuation. So quartz grains are gray and then pore space is black. Then I saturate the core with X-ray attenuating brine. And so now you can see that the black pore space is now filled with this lighter color because it is attenuating X-rays. And I'll show you a video of the injection process. So this is injecting CO2 into the rock. So as I said before, or maybe I didn't, maybe I forgot to say before, full 3D acquisition takes at least 12 hours. And this injection process only takes a couple hours. So we don't have the temporal resolution to visualize the injection in 3D, but we can look at it in 2D. So that's what I'm showing there. So we take a 3D scan after that fluid has been injected, and now you can see there's some regions of the rock that have brine filling the pore spaces and some regions that have CO2 filling the pore spaces. We then do a brine injection. So in this one, the lighter color, regions where there's lighter color, that's brine invading the rock. And regions where there's darker color, that's where CO2 has shifted and moved to fill other regions of the rock. And then we take another scan and another full 3-dimensional scan. And in this scan, now you can see that there are little tiny bubbles of CO2 that are trapped throughout the rock. So all of those little bubbles are capillary trapped. So that's our capillary trapping mechanism actually in action. I have another video here, but I think I might skip it because I'm a little bit short on time. So I want to show you some results from some different kinds of samples. Here we have a couple different layered samples on the left and right, and in the middle I have a quarry sandstone, so it's quite homogeneous. The 3D sort of models of these different samples look like this. So you can see that one of the reservoir sandstone is layered but quite coarse grained, and the other reservoir sandstone is fine grained but also with layers. And then our quarry sandstone looks fairly homogeneous. Can look at the injection patterns in these different kinds of rocks. So in the sort of homogeneous, in the homogeneous system, the CO2 gets fairly evenly distributed by the end of the CO2 injection. Whereas in this layered system, you can see that, yeah, the CO2 mainly fills these large layers, and it takes a longer time for the system to reach what looks like a steady state. And then, of course, we can also look at the capillary trapped CO2. So these CO2 bubbles that are trapped in the pore space after we go through the full experimental process. So you can see again that in the quarry sandstone, we have fairly uniform trapping, whereas for both of these reservoir sandstones, there's regions with high CO2 trapping, but also regions where there's no CO2 at all. And you can also see that the size distribution characteristics of the trapped CO2 bubbles are significantly different. So this has implications for things like surface area to volume ratios of the bubbles, and the capillary pressure that the bubbles are residing at. So we can also look at individual bubbles of CO2 inside these rocks and make very precise measurements of, for example, bubble volume, bubble surface area. We can look at the fluid-fluid interfaces and measure the curvature of those interfaces for individual bubbles. Another thing that I'm very interested in particular is the topology of the CO2 phase. So topology meaning the connectivity of the CO2 phase. So we can quantify how well connected the CO2 phase is by looking at these 3D distributions and looking at things like the number of loops that the CO2 is forming. This gives an indication of how interconnected the CO2 phase is. And we can also see if there are any disconnected regions or features. And then one of the things that we've done with this type of analysis recently is look at how the fluid connectivity relates back to capillary trapping. And what we've seen is that we are able to quantify and measure the connectivity of the fluid of the CO2. And then we can show that if you inject the CO2 in such a way that it's very well interconnected, once water comes back into the rock and mobilizes some of the CO2 away, we end up with very low levels of capillary trapping. But if you can inject the CO2 in such a way that it's very disconnected when you first do the first injection, then most of that CO2 will in fact be capillary trapped even after you do a subsequent brine flooding process. Okay, so I think basically I'm telling you that we're able to quantify some very interesting physical and chemical processes using X-ray tomography and 3D imaging. So everything's great, kind of, sometimes. I want to just point out one potential pitfall of this process, which has to do with our image processing. So because we're working with image data, the processing of the images can have big impacts on the quantification. So for example, in this larger image, to the human eye, I think it looks like a pretty good image. I can make out what's rock and what's pore space. But when we zoom into this very small region, all of a sudden we can see that it can be very hard to distinguish between the different phases when we're looking at it at that scale. And so what this means is that, for example, if I'm trying to measure the size of that particular pore throat at that point, depending on how I decide to label the pore space versus the grain phase, we can end up with different values for the radius of that pore throat. So we could even end up with, there is no pore throat there, the radius doesn't exist, or we could end up with a smallish number, or we could end up with something that's 2 to 3 times larger. And of course, coming back to this idea of trying to be able to predict, capillary trapping levels, trying to figure out the radius of those small constrictions is very important, and that could have big impacts on what we actually, what we might predict would happen, which makes it more important to actually go through the full experimental series and not just base your predictions based on one image. Okay, so I'll just summarize here. We, and I mean that as like the global we, need to be developing emissions mitigation and also negative emissions technologies. And of course, this requires additional investment into research into renewables and other energy sources as well, if we want to be able to keep warming to reasonable levels. I think it's very likely that geologic storage will play a role, at least in the short term and likely in the medium term, among these emissions mitigation and negative emissions technologies that we need. And in order to accurately predict CO2 movement and then design safe injection programs, we really need to pay attention to the small scale details. So that includes in terms of things like geology and also fluid mechanics and dynamics. And we also need to pay attention to the details of these analytical techniques as well. Yeah, I guess I'll just have one more comment, which is I do think this is a really big problem. I think it's one of the biggest problems that we face, and it requires a lot of different knowledge in a lot of different spaces. We need to have understanding of fluid physics, of geology, chemistry, and also to look at these problems, we need to understand things about X-ray physics, and also there's a lot of computational aspects to all of this. So all these skills are really necessary in order to try to tackle this really significant problem. And I guess to that point, we are recruiting PhD students to work on some of these problems in our training center. So if any students out there are interested in PhD programs, I encourage you to check out the positions that we have available.